Hi, everybody. This is Jen.
And this is Aisha.
Thank you for coming to today's webinar. Today's webinar is part one of the two-part Small RNA-Seq webinar series. In today's webinar, we're going to talk about introduction, and Aisha and I will co-present this webinar. So by the end of the webinar, we hope you understand the workflow for small RNA sequencing. We hope you understand the key considerations for small RNA sequencing from RNA isolation to data analysis. And coming up on June the 7th, we're going to talk about best practices in library preparation to ensure that you have a successful library prep and get beautiful sequencing data. And the reason we put this two-part webinar series together is based on our customers' needs and feedback. We want you to understand how to plan your RNA-seq experiment, how to get nice, beautiful data from your small RNA sequencing, and know how to analyze your data. All right, so in today's webinar, here's the outline. I'm going to talk about part one, small RNA basics. So we're going to talk about the pathway for microRNA, different types of small RNA, and the small RNA sequencing workflow. And then we'll go into the second part, which is the key considerations in planning your small RNA-seq experiments. I will cover the RNA isolation, library prep, and sequencing. And then my software colleague, Aisha, will talk about the data analysis, and then she will finish off with part three, which is the resources. So without further ado, we're going to start with part one, the basics of small RNA. So you probably heard about microRNA. So let's just revisit how microRNA is generated in the cell. It all starts with genomic DNA. RNA polymerase will transcribe the genomic DNA information into what we call the primary microRNA. Each primary microRNA can contain one or several precursor microRNA. So then it will be processed into the precursor microRNA. The precursor microRNA will be further processed into the mature microRNA. This process is mediated by the Joshua and Dicer pathway in the cell. And because of the processing by Joshua and Dicer, this results in a unique structure of the mature microRNA, that is the 5' phosphate and the 3' hydroxyl groups. So I want you to keep this in mind because that is the foundation of how our library prep kit is designed. And that's a very important feature of the microRNA. We'll come back to that in future slides. So mature microRNA is something our library prep kit and our software will report. Our library prep kit will capture and our software will report also the isomer. You might have heard the term isomer. So what is isomer? Isomer is just a variant of microRNAs that are generated from the same microRNA precursor. Isomers can be generated by 5' modification, shown on the right, or by 3' modifications. It can be also generated by 3' addition or nucleotide substitution. The function of isomers are not all that clear right now, but it is real. Thanks to NGS, we know isomers do exist. They're not just an RNA degradation product coming out of the library preparation. It has been well established among different species in metazoa. So even though we don't know the function, they're there, and they may have important functions, so that's why we capture and report them. We've talked about the small RNA term. Small RNA is actually not a functional group. It simply refers to different RNA species that are just small in size. So there are different types of small RNA. So here are the small RNA types that people study a lot: the microRNA, the small interfering RNA, and the Peewee interacting RNA. What do they have in common? They are all small in size. You can see, anywhere from 18 to a little bit over 30 nucleotides. Another unique feature that they share in common, which is, again, very important, is that they all have the 5' phosphate and 3' hydroxyl group. So this is a key feature because they are processed by the Georgetown dicer pathway. This 5' phosphate and 3' hydroxyl group has also been reported for precursor microRNA. So you might wonder, Illumina has a couple different kits for mRNA and total RNA sequencing. Why do you have a separate kit for small RNA sequencing? That's just because small RNAs, by definition, they're very small. They're too short to be captured by the regular mRNA and total RNA kits. That's why we have a separate kit for small RNA. And we'll talk about how we capture the small RNA in the future slides. So we talk about the different types of small RNAs. How do we capture them? How do we sequence? Here is the common sequencing workflow. We're going to start with RNA isolation, and then go into library prep, and then sequencing, and finish off with data analysis. So in the second part of the webinar, we're going to talk about the key considerations in each of the 4 steps listed here. We will start with RNA isolation. The first question people typically ask is which RNA isolation kit should I use for my sample? The short answer is there's no specific recommendation because which kit to use or protocol to use depends on the sample type. It is always a good idea to check the literature and see what other researchers in your particular scientific field used in the past to isolate RNA. Even though we don't have specific recommendations, we do have guidelines in terms of what to look for in the RNA isolation kit/protocol. First of all, the kit or protocol should give you pure and good quality RNA. Secondly, it should also retain small RNA. Just keep in mind, not all the RNA isolation kits out there on the market retain small RNA. So make sure that when you use, actually keep the small RNA when you isolate the RNA from the tissues or cells. And third, and it's also the most important one, make sure the kit or protocol does not introduce bias during isolation. I'm going to spend a few minutes on this one. The most important thing in small RNA sequencing is to avoid bias. When I say bias, I mean, are all the small RNA species in the cell or the tissue that you're working with retained during isolation, library prep, and it's all in your final library to be sequenced and to be analyzed? That's one thing. Another thing is, are the relative abundance of different microRNA species retained during isolation, library prep? That's the second part of bias. So bias can be introduced at different stages. It can be introduced during RNA isolation. So that's why it is very important to make sure that the RNA isolation kit or protocol you use does not introduce bias. Once we isolate RNA, the first thing that we are going to look at is the quality of the RNA input. The input requirement, quality and requirement for our TruSeq small RNA kit is that you need to have intact RNA. The kit is not compatible with degraded FFP RNA. When we say intact RNA, it means ring value greater than 8 for mammalian species. For other species, you might want to check the literatures and see what the criteria is. How do you QC the quality? We highly recommend you run the RNA on an RNA chip on the bioanalyzer. You can also run a formaldehyde 1% agarose gel and stain with ethylene bromide. Knowing the quality of your RNA is important. And because we're working with small RNA, you can also check the presence of small RNA on the Agilent small RNA analysis kit. This is not required, but it would be good to check the presence, especially if you're new to our RNA isolation protocol or kit. Overall, the input quality is the number one most important factor in RNA sequencing and small RNA sequencing. It's just because the quality of your RNA depends on whether you can use the kit and how much input you start with. Okay, once we verify the input quality, then we're going to measure how much RNA we have. For the TruSeq Small RNA Kit, you can start a protocol with 1 microgram of total RNA. This is high-quality total RNA. You can also start a protocol with 10 to 15 nanogram of purified small RNA. How do you make sure that you accurately quantify your input? Because that's also important. Right? We recommend a fluorometric-based method, such as RiboGreen, to quantify the input. We do not recommend using nanodrop because nanodrop tends to overestimate how much RNA you have. Once we have the input, we're going to go into the TruSeq Small RNA library prep protocol. A question that's asked often is, what RNA species are captured by the TruSeq Small RNA protocol? So we're listed in here. The RNA species have to meet both of the 2 criteria here. You need to have a 5' phosphate and 3' hydroxyl group. It's 18 to about 30 nucleotide in length. So why are we saying this? Let me show you the workflow of the TruSeq Small RNA protocol. So the TruSeq Small RNA protocol starts with the total RNA or purified small RNA fragments. The first thing we do is we're going to ligate adapter to 3' end. That's the end that has the 3' hydroxyl group. So we're going to ligate the adapter to the 3' end. And then we're going to ligate the 5' adapter. Now it's ligating to the 5' phosphate group of the microRNA. Once adapters are ligated, we're going to do reverse transcription and PCR and to synthesize the first strand cDNA. Then we're going to do PCR amplification. This will not only amplify but also add additional sequences that's needed for library prep and cancer generation. And then we're going to do gel size selection. We select a band about 145 to 160 nucleotide on the gel, which corresponds to 18 to 30 nucleotide of small RNA in length. So because the way the protocol is designed, we are going to capture most mature microRNAs. We're going to capture the small interfering RNA, the PIWI interacting RNA, and some precursor microRNA as well. The next question people typically ask is my organism compatible with the TruSeq Small RNA Kit? Can I use the kit to isolate microRNA or small RNA from my species of interest? Well, you just need to ask one question, and it goes back to the basic of the TruSeq Small RNA Kit. The question is, does the small RNA from the organism that you are interested in have the 5' and 3' 5' phosphate and 3' hydroxyl groups? If the answer is yes, then it is compatible. We know all animals have this chemical modification. The plants, same thing, insects, they all have the 5' phosphate and 3' hydroxyl groups. For other organisms, we recommend that you check the literature and see whether the small RNAs from the organism have the right chemical groups. Now we're going to move on to the protocol. I'm going to talk about the key step in the protocol, which is the 3' adapter ligation, and that's also the very first step in the protocol. Remember, we talked about, the most important thing in small RNA sequencing is to avoid bias. Bias can be introduced during the library preparation. So what we did in the TruSeq Small RNA protocols is that we designed the protocol based on the unique feature of the small RNA so that we get the most quantitative conversion from RNA into cDNA and cDNA into the library, this is critical for reducing bias. And how did we do that? We do that by using a modified 3' adapter as well as using truncated T4 RNA ligase. This ensures when the ligation happens, the reaction only goes one way. That is, the 3' adapter ligates to the 3' end of the small RNA. We use this method to capture everything, to retain everything, to not introduce any bias in terms of relative abundance of the different small RNA species. So this is the key step in the protocol. Definitely make sure you follow the protocol exactly, because otherwise there might be a problem downstream. So we talk about the key steps. Let's talk about the key QC point once you have the library. The final library running on the bioanalyzer should have a size from 145 to 160 base pair. We recommend you do this quality check on the bioanalyzer, not only to check the size of your final library, make sure it's correct, also to check for any unexpected peak. You can also use the bioanalyzer to quantify your library. When using bioanalyzer to quantify your library, make sure you add the molarity of all the peaks that you see on the bioanalyzer. Sometimes you may see two peaks, one corresponds to the mature microRNA, one corresponds to other regulatory small RNA. So make sure you add the molarity because all of them will cluster. That's why we recommend that. If you wanted to learn more about how to ensure a successful TruSeq small RNA prep, come to the second part of the webinar. We're going to talk about the best practices. Now we're going to move on to sequencing. So we're first going to talk about coverage, because that's always the first question people ask. What kind of coverage do I need for my experiment? So let's first talk about what is coverage. Coverage means how many times you sequence through a particular position. The more you sequence through, the greater confidence you have about your sample sequence. So let's look at the slide here. There's a reference sequence in the gray shade, that's a reference. And then the one in the blue shade, these are the sequencing reads. So when we align the sequencing reads to the reference, when we see an A to C change, we're not sure, if we just sequence once, we're not sure whether this is a true like SNP or mutation, or this is a sequencing error, or this is just a read that's misplaced here. But if we sequence it through multiple times, then we can say, yes, this A to C is a SNP or mutation. That's the essence behind coverage. You probably hear about X number of coverage when people talk about sequencing. In the RNA sequencing field, we use the number of reads to describe coverage. That's just because RNA species are present at different copy numbers. So when you say coverage, which particular RNA species are you referring to? You probably get, a really high number of coverage for the high abundant expressor, but low coverage for the low expressors. That's why we use a number of reads to describe coverage in RNA sequencing. The more reads you have, the more reliable you're going to have in terms of counting the number of small RNA species, or more confidence in the sequence that you get from the sequencing run. So for the small RNA sequencing, how many reads are needed? The number of reads that are needed are dependent on the application. Right? If you're doing differential expression and working with animal cells, you probably just need about 1 million reads per sample. If you're doing more like a discovery type of project, discovering new microRNA, then you will need about 10 to 20 million reads per sample. These numbers are for animals. Plants will require more. One thing I wanted to emphasize is that these numbers are very general estimates. Please use them as reference and check the literature and see what the standard is or the spec is in your particular scientific field. The next question is, once you know how much coverage you need, then you wanted to know, okay, there are different Illumina instruments. Which one should I use? How many samples can I sequencing run on this particular instrument? So here's the table. The numbers here are done by assuming you need 5 million reads per sample, and then we're going to take the instrument output and divide it by the million of reads. So here we give you the estimated number of small RNA sequencing samples that can be completed in a single sequencing run for mini-seq, MiSeq, NextSeq, as well as HiSeq. The TruSeq small RNA libraries actually are compatible with all Illumina sequencing platforms. We're just here listing some for your reference. All right, once we decide, okay, we're going to run it on this particular instrument, the next thing we need to decide is, are we going to do a single read run or a pair end run? And how long am I going to sequence in each read? We recommend a single read run. There's no reason why you cannot do a pair end run, but that's not needed because small RNA, as the name states, they're very small. They're only about 30 nucleotide long, at most. So there's really no need to sequence from both endings. The read length, we recommend doing a 1/50 base pair run for small RNA libraries prepared with the TruSeq Small RNA Kit. We recommend 1/50 over 1/36 base pair, and that's because this will give us more sequence information for adapter trimming when sequencing longer RNAs. And let me show you a diagram here. So let's assume, your small RNA insert is about 36 base pair long, and we're doing a 1/36 base pair run. So that leaves you with 6 base pair of adapter sequence. When you're doing like a novel discovery type of project with that 6 extra nucleotides, you may ask yourself, is this really adapter or this is part of my small RNA? You're not sure, right? But if you do a longer run, doing a 1/50 base pair run, now you have all these extra sequence. Now you can confidently call, okay, this is adapter. This is not part of my small RNA. See, that's why we recommend doing a longer run for more accurate adapter trimming. Okay, key QC points. Of course, you always want to make sure that you get good quality data, and for ours, you're going to look at percentage of base greater than with Q30 score, greater than Q30 score. The data output, you want to get as much data as you can from the instrument. The data output is dependent on the percentage cluster passing filter as well as the cluster density. And nowadays, with the massive amount of data you can get from our instruments, people always combine different samples and run them in a single lane or single run. So in most cases, it is important to ensure equal sample representation in the pool. So if you pull the samples after gel purification, then you want to make sure that your libraries are quantified according to the best practices and also verify pipette accuracy. If samples are pulled before gel purification, in addition to the quantifying libraries according to best practices and verify pipette accuracy, you can also do sample QC before gel purification. We will talk more about this in the best practices webinar to ensure that you get equal sample representation in the pool. Now I'm going to pass the presentation to my colleague Aisha. She's going to talk about data analysis and resources.
All right, hi everybody. Give us just a second while we switch things over here. So I am Aisha Wallace. I work in the software support group here at Illumina Tech Support, and I'm going to talk to you guys about Illumina's different small RNA analysis options. So to start, we have a few. So we have a small RNA analysis workflow in the MiSeq reporter software on the MiSeq. For the MiniSeq, the local run manager software, we have a small RNA analysis module as well. And then in BaseSpace, the BaseSpace Cloud and BaseSpace on-site, we also have a small RNA analysis app. So I'm just going to kind of go through. They all do basically the same thing, but the apps on BaseSpace and BaseSpace on-site do more than the MiSeq reporter and local run manager options. So all of these options start by aligning your small RNA reads to a set of 4 reference databases, and then quantifying the hits or the number of reads that align to reads in these various databases. The BaseSpace on-site and BaseSpace Cloud options offer more analysis, so they also perform they also identify novel precursors in your data, and they go on to perform differential expression analysis between groups of samples. then finally, they offer a clustering analysis. So they give you kind of an aggregate result for all of the samples you're comparing, not just your pairwise samples. So the MiSeq Reporter and Local Run Manager options are compatible with human samples. The base-based on-site and base-based cloud options are compatible with human, mouse, and rat samples. All right, so what are the small RNA analysis options by sequencing platform? Well, if you want to use the base-based cloud or base-based on-site option, you can use those options with any one of our platforms. But if you wanted to use Local Run Manager, that software runs only on the MiniSeq. And then for MiSeq Reporter, that software runs only on the MiSeq. And if you want to know more about these different analysis options, we do offer analysis reference guides for each one, which I've linked here in this slide. All right, so now I'm just going to go over the small RNA analysis workflow in a little bit more detail. So the overall workflow is we start with adapter trimming. We do an alignment to those 4 reference databases that I mentioned. And then the base-based options will also perform novel precursor discovery, differential expression analysis, and clustering analysis. So first, the adapter trimming option. If you're using MiSeq Reporter or Local Run Manager, the small RNA workflow includes adapter trimming. And this will be automatically performed when the analysis workflows launch. Adapters will be trimmed at the time of FASTQ file generation, so you don't have to worry about it. If you're using one of the base-based options, base-based on-site or base-based cloud, then the app, keep in mind, does not perform adapter trimming. You do need to trim your adapters before you launch the app with your samples. There is an app on BaseBase that can perform adapter trimming. It's called the FASTQ Toolkit app, which I've linked to here. Also, you can use any tool that you are familiar with using to trim the adapter sequences from your FASTQ files. So if you've generated FASTQ files outside of BaseBase and you plan on uploading them to BaseBase, you can first trim them with third-party tools like CutAdapt or Trim-O-Matic. You can also use the BCL to FASTQ conversion software to trim adapters, so you have a lot of options. So after we trim adapters, the next step is alignment, and we're going to align our reads to 4 databases of sequences using the bow tie aligner. So we're first going to align to a set of contaminant sequences, which is going to be mitochondrial RNA, Phi-X, poly-A, poly-C, and ribosomal RNA. Next, we will align to a mature microRNA database that comes from MirBase. And this is where we identify our known mature microRNAs in the samples. Next, we align to a database of other small RNA, and these sequences come from the NCBI nucleotide database. And here we identify isomers, pRNA, lincRNA, tRNA, snRNA, SNO-RNA, different types of small RNA. And finally, we align to the reference genome. So the alignment order is actually important. So we start with aligning to the contaminants database. Anything that aligns to the contaminants database is assigned as a contaminant, even if it can align to something else in another database. And it's the same with the mature microRNA database. If it doesn't align to contaminants but aligns to mature microRNA, even if it aligns to other or genome, it's considered a mature microRNA, and so on. So the output of the alignment is several text files. And these files just list the hits or the number of reads that aligned to each of the sequences in these various databases. There's several files, so I'm not really going to go over them, but if you see the appendix in your slide deck, I do list some of the key files, but for a full list of the files, check out the analysis workflow guides for the analysis option that you would choose to use. The next step is novel precursor discovery. So at this point, only the base-based options, base-based on-site and base-based cloud, will perform this step. So what we're going to use is a tool called MIRDeep Star actually, which is an integrated microRNA identification tool. It's widely used to quantify known and novel microRNAs from small RNA-seq data. So we'll start by pooling all of the reads that align to reads in the other database in the alignment step. And then we'll take those and use those as our candidate novel precursors. And we'll use MIRDeep, which will predict novel precursors based on the genomic locations of those alignments. And then the next step we're going to do, again, just space option, is differential expression analysis. So for this, we're going to use a popular differential expression analysis tool called DESeq2. And so what we're going to do is we're going to perform pairwise differential expression, so differential expression between pairs of groups of samples, and we're going to compare differences in the expression of microRNA families, precursor groups, known and novel, microRNAs, known and novel, and pRNAs. And like I said, this is a pairwise analysis, so this will be performed for each pair of groups in your data set. The last step is the clustering analysis, and this is kind of our global analysis. So this isn't just looking at groups or samples within a group or samples between two groups, but it's looking at all samples that we've input into the analysis. So the output of this, we're going to get a sample correlation is one of our outputs, as you can see here in this slide. And one thing I just want to point out is just notice that in the top right corner it shows microRNA family. There's actually a drop-down list here, so we can actually look at microRNA families, precursor groups, microRNAs, and pRNAs here. And then when we select the different tabs, we get the different outputs. So one is sample correlation. We'll also get a PCA plot showing how similar or different our samples are to one another. We'll get an expression heat map as a result of the differential expression analysis. And then the last tab, which I'm not going to show, these are our pairwise differential expression analysis results. And this will just show that the results of the pairwise differential analysis tests performed by DESeq and whether or not the difference in expression was considered significant by the software. So among the different output files you get for each sample in your analysis, you're going to get a report. So you're going to get this small RNA sequencing report for each sample, and it contains a lot of statistics. So it will give you information for this particular sample on the small RNA length distribution, the read distribution, so the percentage of reads that align to each of our different databases. And then we'll get a breakdown of the reads that align to those different databases. So among our abundant reads, we'll get to see how many align to the different types of sequences within that database. The same for the microRNA database and the other RNA. We'll also get a list of the top sequences found for each category. So for our mature microRNAs, we get the top 10 most abundant reads listed there. And we'll get the same for our novel mature microRNAs, any novel mature or precursors that were found, our precursor groups, etc. So there's a lot of information, a lot of statistics, a lot of stuff reported in these files. I'd take a look at a couple of them to see what's all in there. And then there's also going to be an aggregate summary report. So this is a report that lists our global analysis results, and it can be very large. For the data set I was working with, it was 50-something pages, so I'm not really showing all of the pages here. So I just want to kind of give you a summary of what's in this. You'll get aggregated hits by category, so you'll have each one of your samples listed, and it'll show you the percentage of reads that align to abundant microRNA, other small RNA, and then a breakdown of hits within that category, and it gives you a nice comparison of all samples together. You'll get your sample correlations like before. You'll get PCA plots, more expression heat maps. You'll get the top differentially expressed species for each of your pairwise analyses. A lot of information reported, but that is basically the workflow. You align, you count the hits to each reference sequence, novel precursor discovery, and differential expression analysis if you're using the base space options. And that's all I have for the analysis part of the webinar. So moving on to the resources, part 3. We have some resources for you on the web. I also want to point you to some of our support webinars and our support bulletins. So first, the resources on the web. On the Illumina website, you can find a support page for the the Small RNA Sequencing Kit, as well as RNA sequencing in general. There's a page that compares sequencing platforms, if you're not sure which platform it is you'd like to perform your analysis on. There's also app user guides linked here as well for the different small RNA analysis options that we provide. And then finally, you can find a list of publications on our website. If you click on that All Publications link, could be helpful in your experimental design. Then we also have a list of the resources used by the analysis tools that I mentioned. So for alignment, we use the Bowtie Aligner, and if you want to learn more about that, you can click on the link here. The base analysis options also use MirDeep and DE-Seq2, so if you want to know more about those tools, click on those links. And then finally, our support webinars. So this is the first part of a 2-part Small RNA-Seq webinar series. My colleague Jen, as she mentioned before, will be presenting on the best practices for small RNA sequencing. And also, there is a 4-part RNA-Seq webinar series that has been recorded, and you will be able to find links, or you'll be able to find those resources on our website as well. If you click on the link, this series focused on mRNA, total RNA library preparation and analysis. And links to our upcoming webinars and our recorded webinars are found on this slide as well. And then finally, our support bulletins. So we have a lot of really useful bulletins written by people in tech support and maybe some of your field scientists. Lots of current information can be found here. So if you click on these links, it'll take you to our support bulletin site. I would urge you to check that periodically. There's always really useful information found there. To find all support bulletins, just click on the link on that slide. And finally, just a reminder that we have expanded our phone hours for tech support in the Americas. We're now open from 5 a.m. to 5 p.m. Pacific time.